Our first speaker is Varun Ullanat. Varun is an undergraduate student at the RV College of Engineering in India. And Varun's talk is about the transcriptomic analysis of the brains of symptomatic and asymptomatic Alzheimer's patients. So the stage is yours, Varun.
Yeah, thank you. Can you see my screen? Okay, cool. Yeah, so good evening, everyone, or good morning, or good night, or good afternoon, wherever you are. So my name is Varun, and I'm an undergrad student in RV College of Engineering, India. My poster, as you can see, is titled Transcriptomic Analysis of the Entorhinal Regions of Symptomatic and Asymptomatic Alzheimer's Patients. So for those of you who aren't familiar, asymptomatic AD is a condition where patients have intact cognition. It is a condition where patients have intact cognition, but neuropathology similar to AD. Asymptomatic AD patients are highly likely to develop AD as well. So we can think of it as an intermediate disease state between the normal condition and AD. The intuition behind this project is that understanding the molecular mechanisms that govern asymptomatic AD could help us in early detection and treatment of a patient before AD symptoms develop. Yeah, so this is the basic workflow. We first took gene expression count data and again some basic pre-processing such as removing batch effects and removing the bad quality probes were done. The data is then split into three study conditions: AD versus asymptomatic AD, as well as control, and asymptomatic AD versus control. Next, we performed a differential gene expression analysis using statistical tools and identified genes with a p-value of less than 0.05. We simultaneously performed co-expression studies. Then we overrepresentation analysis and gene set enrichment analysis were done to identify the significant pathways and gene sets. Finally, protein interaction networks were constructed with the co-expressed genes. So moving on to the results. The first slide is for AD versus asymptomatic AD. We observed that in the volcano plots of the two conditions, of the two cases, most of the genes are overexpressed in the disease condition. Next, again, this is a table of the top 5 differentially expressed genes. In the protein interaction network, so each module actually represents genes which are similar, which have similar expression profiles or similar co-expression. We can see proteins of genes such as SNAP25 and FOS highly pronounced as hubs in these networks, and these are the classical genes related to AD. So moving on to the AD versus control case. So this is again very similar to the first case. Again, most genes are overexpressed, as you can see in the volcano plots. So finally, the last slide is for the asymptomatic AD versus control case, the one we're actually interested in. So again, most genes are overexpressed as compared to healthy individuals, but not as much as the AD conditions. We only obtained about 40 differentially expressed genes here, and again, the top 5 are given. Interestingly, we obtained 5 different modules for the asymptomatic AD case, 5 different protein interaction networks, suggesting that multiple gene sets are involved. In module 4, you'll notice APOE4, a gene synonymous with AD as hub. So studying the other genes involved would explain, or to an extent, explain how APOE4 works. Next, so these are basically the Venn diagrams of the number of differentially expressed genes in each study condition. An interesting observation is that there is no single unique gene involved in asymptomatic AD. Also, about 5,000 genes are common in the AD versus asymptomatic AD and the AD versus control case. The Venn diagram on the right is of the significant pathways we saw during our analysis. We observed that about 17 pathways are unique to the asymptomatic AD versus control condition. So finally, the table to the left shows the classical perturb pathways known through literature and, of course, observed in our study. Again, the table to the right shows the significant pathways we observed in the asymptomatic AD versus control condition. Again, we saw several interesting pathways. So innate immunity pathways and neutrophil degranulation were shown to be affected, which indicates that there is an immunological aspect to asymptomatic AD onset as well, similar to AD. Coming to the conclusions, so again, it's consistent with the previous findings on genes and pathways involved in the onset of AD. Again, so the genes such as APOE4, FOS, SNAP25, and STC4 which have been shown to have altered expression levels in AD patients are highly pronounced in the current study results as well. I mean, with some further analysis, we actually identified three features or three salient features we observed in most of the protein interaction networks and the significant pathways. So first, we observe SIT1 as an interaction hub in the protein interaction networks of all cases. So further analysis showed that SIT1 is involved in a pathway that leads to A-beta plaque generation, which is an important biomarker of AD. Secondly, we observe STC4 as a co-expression hub in all the cases as well. So by doing a similar analysis, we discovered that this gene is also involved in A-beta plaque generation. Finally, we observe CYBB as the most differentially expressed gene in symptomatic AD condition. So this gene is known to be involved in multiple sclerosis, but the function in AD is still being researched. Again, so a similar analysis was done for the temporal, frontal, and the cerebellum regions of the brain as well. But we couldn't actually include it in the final presentation because of constraints of time and space. Yeah, so thank you.
Thank you very much. That was a great talk. Thanks a lot. So are there any questions? You can share your questions on the Q&A section. Also post your questions on the chat part as well. So for now, I'm not seeing any questions. Oh, someone has raised hand, I think.
Hello.
Hello.
Hi, nice talk, thanks a lot. I have one question for the gene that was most differentially expressed in the asymptomatic AD. So could you talk a bit about the function of that gene, or speculate on the role maybe for the outbreak of AD?
So CYBB is, I think that was the one which I mentioned,
Yes.
is actually, it provides instructions for making a protein called cytochrome B245 beta chain. So it's actually it's one subunit of a group of proteins that form an enzyme complex called NADPH oxidase, which again plays an essential role in the immune system. So again, we know that AD has an immunological aspect to it because of the degradation in the extracellular matrix and such. So is possibly the reason why it's differentially expressed.
Cool, thanks. That's very interesting, yeah.
I actually have another question about cytochrome B. So I think I remember hearing about it also in the context of mitochondria, right? So it's like a mitochondrial protein, and in AD obviously you have like mitochondrial dysregulation. Could that be might that be what's dysregulated or somehow linked?
Yeah, I mean, it could be, but I think the primary reason is this, but I'm not really sure. I'm primarily a bioinformatician, so I don't want to talk about, exactly the molecular mechanisms in AD. So this is what we found in our analysis, I'm just sharing what we found. Yeah. So again, you can do further studies to check.
Yeah.
Thank you for the questions. We have another question, and I guess I need to ask that. So this is from Eva Limberopoulos. I hope I pronounced the name correctly. How do you interpret the significance of finding perturbations in innate immunity in asymptomatic AD?
Yeah, so like I mentioned before, immunity is, it's well studied. So in Alzheimer's disease, in the actual Alzheimer's disease, not the asymptomatic one, again, so immunity is a very important component. We see a lot of genes and a lot of pathways involved in immunity to be perturbed. Again, so if I can show you the slides, actually I didn't mention the immunity part. In the AD, table, the last table, which was there in the slide, it was mostly elastic fiber deformation, such things like that. But we did find a lot of pathways protrude in the immune system as well, relating to AD, and it's been well studied. So again, it seems plausible that it is obviously affected in asymptomatic AD as well, because it's one of the reasons why the AD progression actually takes place. So it's like the immune cells start destroying the ECM and the ECM of the nerves. And yeah, so I think that's what she meant. I'm not really sure. The question again, I don't want to dive deeper into or speculate about why it's actually integral in AD or asymptomatic AD progression.
Okay, thank you. Thanks a lot. So yeah, I guess we have no other questions for this talk.
Okay.
Yeah, so I would like to thank you again for the wonderful talk, and also thanks for the excellent questions. Was very informative. So we can move on to our